The only job of sperm is to find the egg and deliver their DNA package, right? Wrong. Turns out they do a lot more than just that. They also contribute things that can affect the phenotype or the characteristics of the resulting child beyond just the genetic makeup or the DNA. In this video, I'm going to briefly discuss this really interesting paper titled Epigenetic Inheritance of Diet-Induced and Sperm-Borne Mitochondrial RNAs. Now, this study was done in mice, and what they set out to find was what are the impacts of a high-fat diet on not only the sperm, but the resulting offspring of the mice on this high-fat diet. And more specifically, they wanted to see where the impact on the sperm is happening. There are essentially two stages to sperm development in mammals. Step one is carried out in the testes. This is the development of sperm cells from spermatogonial stem cells. So they go from this little round cell into, a head and tail as a sperm, like the sperm as we know it. Stage two of development, the sperm travel to this structure called the epididymis, which is located right on top of the testicle. And in the epididymis, sperm go through final stages of maturation where they gain the ability to be motile or swim. And for many years, we kind of thought that's all that happens in the epididymis. But in recent years, what we've been finding is the epididymis is contributing more factors to the sperm after they've been developed. And some of these really interesting factors are these group of molecules called small non-coding RNAs, which are involved in epigenetics. Epigenetics relates to factors that can regulate or control the expression of the genes in your DNA. I like to think of them as like a light switch. It can turn genes on, it can turn genes off. It can also be a dimmer switch where it lowers the gene expression or increases the gene expression. What we've learned about the epididymis is that on top of just going through the final stages of maturation to gain motility, there are also these things called epididymisomes that come from the epithelial cells lining the epididymis. These epididymisomes are essentially these little packages that go and fuse with the sperm and deliver more components into the sperm cell. And a lot of these components are these small non-coding RNAs or epigenetic factors. And these epigenetic factors after fertilization get into the egg and then have an effect either on early embryonic development or in the phenotype of the resulting offspring. Super fascinating stuff to me, especially as an embryologist. I mean, we're so hyper-focused on DNA and DNA fragmentation and DNA quality. Why are we talking more about these non-coding RNAs? So back to this paper. What they wanted to see was, does a high-fat diet impact sperm? Does it impact these epigenetic factors? And if it does, is it happening in phase one in the testes or in phase two in the epididymis? Basically, what they did in the study was they fed 6-week-old male mice high-fat or low-fat diets for 2 weeks. Then they further split them into two groups. Group one, after the two-week diet, they were allowed to breed and then they monitored the offspring. Group two, they took them off of the high-fat diet. They let them mate in order to just, clear the pipes. And then after 4 weeks, they allowed them to breed and then they monitored the offspring. The reason for the 4-week gap is that sperm take a certain amount of time to develop in the testes, and that varies from species to species. In humans, it's about 70, 75 days. Whereas in mice, I think it's around like 35 days. That's why they did 4 weeks. So by waiting 4 weeks off of the high-fat diet, then that would see if there was an effect on the sperm that were being developed in the testes versus the ones that mated right after 2 weeks. That would be seeing if there was an effect on the sperm that were already in the epididymis. Now, what they found was that the high-fat diet did not have an impact on the spermatogenesis, the phase one in the testes. And they say so here, spermatogenesis and male reproductive fitness are not affected by the high-fat diet feeding, as shown by the testis histology and the diameter of the seminiferous tubule, I can never say that word, sperm motility, and the rates of successful fertilization and pre-implantation development. Transcriptomic analyses of round spermatids and testis show normal spermatogenesis and little to no high-fat diet effect. Translation: if this were a semen analysis done on a human, they would have found that all parameters were normal. The semen analysis would have come back as normal. Concentration, motility, total count would have been normal. Please, if you've heard this part and you think, oh, a high-fat diet, no effect on my sperm, I'm turning this video off. Don't do that, because there is an actual effect on the sperm. They're just showing it's not happening in the testes. When they looked at the offspring, what they found was the offspring didn't have a higher body weight. However, 30% of the males presented with insulin resistance and glucose intolerance. Insulin, you need it. That's what helps your cells to uptake glucose and convert it to energy. I'm no physiologist or nutritionist, but insulin resistance is a bad thing. It can lead to multiple other problems like obesity, cardiovascular disease, PCOS. The fact that it was male mice that were having this insulin resistance, the paper, as far as I could tell, didn't come up with a reason why it would be male and not female inheritance. All they said was that was kind of interesting that it was in the male group. And while this study was done in mice, they brought up the fact that we see similar things in humans. Using gene expression and genome-wide association study data from children included in the Leipzig adipose tissue childhood cohort, we found that about 30% of the high-fat diet intolerant signature genes are also expressed in human adipocytes and associated with childhood obesity. Furthermore, they talk about how parental obesity, especially maternal, is the strongest risk factor for early onset obesity in children. However, paternal obesity is also independently related to offspring BMI and issues with obesity. In families with lean mothers, paternal overweight doubles offspring obesity risk, which is further worsened by paternal obesity. So not just being overweight, but being obese can significantly increase the odds of the offspring also having obesity risk. So back to the sperm. For them to see that this high-fat diet isn't impacting spermatogenesis, it's not affecting the way the sperm development, and it's not affecting the genes, the question then is, why are these mice developing insulin resistance? What they found was the key is transfer RNAs. If you remember back to biology 101, transfer RNAs are involved in the translation of messenger RNAs. They deliver the amino acids that are being built into the chain of a protein. We have nuclear transfer RNAs that are found in the nucleus of our cells, but there are also mitochondrial transfer RNAs. If you remember, mitochondria have their own DNA. As such, they have their own mitochondrial transfer RNAs. In the mice that were given this high-fat diet, what happened was a downregulation of the tRNAs in the nucleus and an upregulation of mitochondrial transfer RNAs. This upregulation also translated into an increase of transfer RNA fragments. This is something that's genetics tends to go over my head a bit. I don't really understand what these transfer RNA fragments are. Suffice to say, there is an increased amount of these mitochondrial transfer RNA fragments in the sperm as a result of the high-fat diet. After fertilization, these mitochondrial tRNA fragments are getting into the egg. And this is something that also is hard for me to comprehend because the sperm is like 1,000 times smaller than the egg. But there's enough of these mitochondrial tRNA fragments that are getting in there, and then they are having epigenetic effects in this newly developed zygote. And these epigenetic effects are the fact that the offspring are then becoming insulin resistant and glucose intolerant. So to put that all into a little more simple English, a high-fat diet was stressing out cells and resulting in an increase in these mitochondrial transfer RNAs and these mitochondrial tRNA fragments. These were being delivered into the sperm via these epididymosomes in the epididymis. After fertilization, these mitochondrial tRNA fragments then go about and essentially wreak havoc in about 30%, and in these mice, 30% of the male offspring. On top of affecting insulin resistance in the offspring, there was also an effect on the early embryonic development, which as an embryologist, like I said before, is something that's very interesting to me. This part in particular, it talks about how as embryos, fertilized embryos, move through the oviducts, they're exposed to different concentrations of oxygen. Mammalian pre-implantation development represents an example of continuous metabolic adaptation in which developing embryos face a gradient of oxygen concentration from the oviduct to the uterus, resulting in a six-fold increase in oxidative phosphorylation in blastocysts compared to cleavage stage embryos. Oxidative phosphorylation is what happens in mitochondria. Premature activation of oxidative phosphorylation during preimplantation development leads to alteration in embryo ultrastructures, mitochondrial structure and function, and glucose intolerance in adult mice. And paternal diet can influence preimplantation developmental timing and early embryo metabolism. Although the developmental timing of these affected embryos is maintained, their gene expression profile suggests premature activation of oxidative metabolism associated with paternal overweight and inheritance of these mitochondrial tRNAs. As I translate that into what we see in the embryology lab, this is something we would not probably notice. The embryos are still going to develop to the stages on time. However, there are some serious metabolic issues going on in these cells as a result of a high-fat diet. The biggest noticeable effect, obviously, is that offspring run a higher risk of obesity and having insulin resistance and glucose intolerance. So, moral of the story, moral of this paper is, one, very interestingly, the epididymis is in a very real way sampling the environment, the current health status of the male, very close to reproduction, very close to the time of ejaculation and fertilization of the sperm with the egg. And this sampling of the environment and health status of the male is being transferred to offspring, not via genes, not via DNA, but through these epigenetic factors. So application to you, if you are trying to get pregnant, it's a good idea for the male partner to get on top of their health. Please know this is not me shaming you or guilting you or having any kind of judgment on you, I promise. This is more just if you were looking for data, scientific reason, backing up why, getting on top of your health is a good idea if you're trying to get pregnant. Here, this is it. This is a good reason why. And hopefully it's encouraging and not shaming. Cat. This paper also does not provide any kind of information on this is what you should do in order to improve your health status. The purpose of this paper was to investigate where is the effect of a high-fat diet on sperm and the resulting offspring, and what are the mechanisms that are explaining how that's getting passed down. So as an embryologist, I'm not going to pretend to be a nutritionist and give you advice. I think there's just about infinite amounts of resources out there. Everybody's talking about health and diet and nutrition. So hopefully this just gives you a little bit of more of a motivating factor as to why getting that in order in your life is a good idea. Me and Reggie, thank you for watching. And he says that he really likes eating a lot of cat food and that he will not help you with your nutrition either.